Okay, so we were talking about color blindness because it's a sex-linked trait, and today's big theme is sex-linked traits. These are non-Mendelian traits that we're covering right now. The unit that we're currently in, or the learning target that we're currently under, is learning target H3, the non-Mendelian traits. When I say non-Mendelian traits, I mean it's traits that don't really obey the normal basic Punnett square rules. They're traits that are weird compared to what Mendel figured out. Remember, Mendel figured out the basis of all of genetics. That's why Mendel gets called the father of genetics, is because 90% of how genetics works is just what Mendel figured out. What Mendel figured out was that parents carry 2 copies of every gene. When they go to make a sex cell, they pass on one of their 2 copies. That's why we do Punnett squares the way we do. That's what Punnett squares are supposed to represent, is a parent carrying 2 copies of a gene, but only passing one or the other onto their offspring. And both parents do that. That's why a basic Punnett square does a pretty adequate job of explaining how Mendelian genes are passed. Now what we're talking about is non-Mendelian genes, like sex-linked traits. Non-Mendelian genes are going to need a special modified Punnett square to figure out their odds. So let's start with that. We talked about what a sex-linked trait was last week. Now we're going to look at how you would actually pass them to your kids. So I was telling you last week, I was colorblind. I have a sex-linked trait that I got from my mother. I also have 2 children. I can guarantee that neither one of my children are colorblind. But I also know that 50% of my daughter's boys, if she had kids someday, which she won't, but if she had kids someday, my daughter would pass it on to 50% of her boys and none of her girls. I can tell all that because I know how sex-linked traits are passed. So you should know that too. Let's talk about it. So let's do a practice problem with sex-linked traits. Let me be perfectly clear, because no matter how many times I say this, there's somebody that doesn't listen to this next little tidbit, and they try to do sex-linked trait problems, try to do sex-linked trait setup with a non-sex-linked trait problem. You'll get the wrong answer. This is a different type of Punnett square that you would only use if I said in the problem very clearly, this is a sex-linked trait. If I don't tell you it's a sex-linked trait, don't use a sex-linked trait Punnett square. You'll get it wrong. Okay. Color blindness is a disease that causes you not to make normal cone structures in your eyes. That's what's wrong with people with color blindness, is that the physical structures in their eyeballs are built wrong. I don't make the correct proteins to be able to pick up all the wavelengths of color. It's a recessive gene carried on the 23rd set of chromosomes. And when things are carried on the 23rd set of chromosomes, it gets weird, because the 23rd set is not like every other set of chromosomes you have. You have 2 number 1s, 2 number 2s, 2 number 3s, 2 number 4s, 2 number 5s, all the way up to 22. Chromosome pair number 23 doesn't necessarily match for everyone. Who doesn't have a matching set of 23rd chromosomes? Me. And everybody else who's a male. Males don't have matching 23rd sets of chromosomes, because males have one X chromosome, but then they've got a non-matching Y chromosome with different genes on it entirely. Girls, on the other hand, have 2 X chromosomes that have matching genes, possibly different alleles for those genes, but they have a matching set just like all their other chromosomes. So for girls, they have 2 number 1s, 2 number 2s, 2 number 3s, 2 number 4s, all the way up through 23. Their number 23s match too. So if they get a broken number 23, they get a backup copy from the other parent. Boys don't get a backup copy for number 23. So if I get a broken number 23, I'm screwed. That's it. It's broken, and I have to read that one because I don't have a backup copy. I have to read the busted number 23. So that's what happens with boys with sex-linked traits. Boys are more likely to show the trait in their phenotype because they don't have a second chromosome that could possibly be carrying a normal version of the gene to cover up the effect of the bad gene. So here's my question. What are the chances that a couple will have a boy with a disorder if mom is a carrier, she's heterozygous, and the father does not have the disease? Couple is will have a boy with a disorder if mom's a carrier. In other words, she's heterozygous, how we would explain it if it was a normal Mendelian gene, and father doesn't have it. So let's take a look at how this works. I'm going to try something tricky here and see if I can do it. Oops, I broke it. Hold on, I'm trying to draw things, but I'm not good at drawing. Give me one moment. Can I do this? Can I do it with fill? No, I can't. They give me a fill tool? No, they don't. Never mind, I'll just have to do it this way. Okay. Mom's a carrier, dad does not have the disease. So here's how we're going to do this. Maybe. All right, I got to switch over to a whiteboard. That'll work. Here's how we start this problem. This is only for sex-linked traits, so make sure you understand this is a sex-linked trait. This is a sex-linked trait example. This is only for this type of Punnett square that I'm going to show you is only for sex-linked traits. So first, we start by doing what you've always done for all of these problems. We're first going to start by drawing a Punnett square. Usually, once we've drawn the Punnett square, we'd put the 2 copies of each gene that mom carries and dad carries on the top and the side of the Punnett square. This time, we're not going to do that. Because with sex-linked traits, it's not just one gene that matters. It's one entire set of chromosomes. So instead of just genes, I'm going to put chromosomes on here. So let's put dad up on the top here. Dad, for his 23rd set, is going to have this going on. He'll have one X chromosome that I'm going to represent with an X, and he'll have one Y chromosome that I'm going to represent with a Y. Yeah, we said dad was normal. This is my parents making me, okay? So here's my dad. He's got a normal X chromosome, normal Y chromosome. Here's my mom. I know she gave me color blindness because my grandpa was colorblind on my mom's side. That's where she got her busted X chromosome from. So she's got, my mom is my mom, that makes her a girl. She has 2 X chromosomes for her 23rd set of chromosomes. Again, Punnett squares for sex-linked traits, only for sex-linked traits. The Punnett square is going to be different because we're putting whole chromosomes on the top and on the sides here. The boy gets an X and a Y. The girl gets an X and an X. Now, I know that one of my mom's X chromosomes has the broken color blindness gene on it. I'm going to mark that broken X chromosome with a C on it to represent colorblind. So this is a normal X chromosome. This is a X chromosome that carries the broken gene for color blindness on it. Okay, my mom's not colorblind. Why? Which one does she read? Yeah, she's her whole life, all the cells in her body read this X chromosome that's not busted. So she reads this normal one, makes normal eyeball structures, and doesn't have color blindness. Good? So her phenotype is no color blindness. All right, now let's have my parents make babies. Gross. You'd never want to think of your parents doing that, but there they go. Ew. No. Okay, since I'm a guy, I got a Y chromosome from my dad. I also get an X chromosome from my mom. Oh, the only one I could get from my mom there. Oh, there it is. Let's take a look. Let's interpret this Punnett square now. Okay? Watch what the results end up being. Right here, what kind of kid is that?
A normal child.
That is a normal child of what biological sex?
Girl.
That is a girl kid, right? She got an X chromosome from mom, X chromosome from dad. You got 2 X chromosomes. The biological sex of that kid is a girl. Yeah? All right, this one. Got an X chromosome from mom, got this Y chromosome from dad. What kind of kid is that?
Normal boy.
That's a normal boy kid who's not colorblind. He got a normal X chromosome from his mom, yeah? Good. Here's the next kid. Got X chromosome from mom, or from dad, and got an X chromosome from mom. Did end up with 1 broken X chromosome. What kind of kid is this? Boy or girl?
It's a girl.
It's a girl. Is this girl colorblind? No, because she'll read the good X chromosome, ignore the broken colorblind X chromosome. Okay? And then finally we got this kid. He's got an X and a Y chromosome. That makes him a boy. He's got the busted X chromosome. Does he have a backup X chromosome to read? No, so when he goes to make eyeball cells and he needs eyeball proteins, he has to read this busted DNA on this X chromosome. He's going to build his eyeballs wrong because he only has this X chromosome to read. So, he builds broken ones. What are the odds that I'm going to end up with a colorblind kid? Yeah, 1 out of 4 chance. Of colorblind kids. Or you could think of it as half of the boys. So of their total kids, it's a 1 out of 4 chance that they'll have a colorblind kid. It's half of all of their male children. Half of all their male children will end up being colorblind. If this was a normal trait, it wouldn't work that way. So if we work this using the wrong Punnett square, look what would happen. If I work this like a normal Punnett square and said colorblindness was, let's say that dad was homozygous recessive and mom was a carrier. So she's got big C, little... no, I'm sorry. I screwed up the setup of this. Let's say dad's got no history of it. He's got no problems with colorblindness. He'll be homozygous dominant. We're going to say the recessive gene causes colorblindness. If mom was a carrier and it was a normal gene, it would look like that. And if we did this normal Punnett square, again, this is the wrong way to do this problem because I told you in the setup of the problem it was a sex-linked trait. We'd end up with that Punnett square. If it's dominant to not have the disease, does this kid have it? No, this kid? This kid? No, this kid? No. So if it wasn't a sex-linked trait, there would be no way this couple could make a colorblind kid. But it is a sex-linked trait, so that's not the right odds. You use the wrong kind of Punnett square if you tried to set up normal Punnett square and don't do that. Like, don't use a normal Punnett square to set up a sex-linked trait problem. The only time you need to use one of these modified Punnett squares where we bother drawing out the entire chromosomes is if it's a sex-linked trait. Don't use a sex-linked trait square on a non-sex-linked trait. You'll just get it wrong. Do we get this? Kinda? Question.
Wouldn't this technically mean that males show colorblindness more than females?
That is exactly right. Males show colorblindness far more often than females. Doesn't make it impossible to have a colorblind female, but you'd really have to work at it. Okay? It would be very difficult to make a colorblind female. That's why, on average, it's a little over 6% of guys have colorblindness, and it's like 0.000 something % girls that have colorblindness. So that's odd. Like, it's pretty common. 6 out of 100 guys you meet are going to be colorblind. Whereas, like, you'll virtually never... I've had one, in all my years of teaching, I've seen literally thousands of students. I've had one colorblind girl. That's a lot of people, and only one of them is colorblind. I've had tons of colorblind guys. Like, lots and lots and lots of them. Cool, good. So just because it's carried on that 23rd set of chromosomes, it affects how often we see the trait in the population. That's kind of interesting. Like, I don't know, it's a little weird. And there's diseases that are carried on the 23rd chromosome that are far more serious than colorblindness. Like, colorblindness is minorly annoying, and like, sometimes I buy like shirts that don't match well and stuff. And like, I don't know, it's not that big a deal. Like, I don't think it's ever really hurt me in any way. Besides, little kid school didn't go well when everything was like color-coded. I don't do color coding. But anyway, colorblindness isn't that big deal. But stuff like hemophilia, the most common type of hemophilia, that's a sex-linked trait carried on the 23rd chromosome. Hemophilia is a disease where your blood doesn't clot correctly. So if you get a minor cut or something, it just bleeds and bleeds and bleeds until you die. Like, it's a pretty serious disease. Hemophilia is carried on the 23rd chromosome. Far, far, far more common in guys. Barely even happens to girls ever. And again, it's because girls have 2 X chromosomes and it can always read their backup copy. Guys don't have that option. We don't have a backup copy for our X chromosome. Are we happy with this? That's a sex-linked trait. Let's see here. What else do I got? Now let's say that this boy with colorblindness grows up and wants to have kids with his wife. You know, wife's father had the disorder, but she doesn't. What are the chances that their boys will have the disorder? What are the chances the girls will have the disorder? All right, let's do this one. Again, still a sex-linked trait problem because color blindness is a sex-linked trait. So now let's do this same scenario with another Punnett square here. Make sure you're following along so you understand how to do this, because sex-linked trait problems are a little bit different. Step 1, I saw what are the chances, so that always tells me draw a Punnett square to figure out chances. And again, since this is a sex-linked trait, and we're only using these type of problems for sex-linked traits, I'm going to put the guy's genes here Or actually, I'm not putting genes, I'm putting whole chromosomes there. He's a guy, this is dad, he's got an X chromosome and a Y chromosome. I said the boy with color blindness. So this is a boy. If he's got color blindness, we're going to mark that little X chromosome that it's got the color blindness gene on it. I'm going to put a little C by it. All right?
You always have to use the letter C? Or
You can use whatever letter you want. I just use C because we're doing color blindness. If I was doing hemophilia, I'd put a little H by it. It doesn't, but you don't have to do a cute little abbreviation. You really can pick any letter you want. It's just so you can keep track of where the gene you're interested in is going. All right, then I'm going to put the girl over here. What kind of chromosomes would a girl have for her 23rd set? 2 X chromosomes. X chromosome, nya. X chromosome, nya. Okay? Remember I told you, though, the wife's father had it. Well, this girl got 2 X chromosomes. One of her X chromosomes came from her... One came from her dad, one came from her mom, right? And if we're saying the dad had it, dad only has 1 X chromosome to give. So if dad had a girl, one of her X chromosomes must have come from him. And if he's colorblind, he only had busted X chromosomes to give. So we know this girl must be carrying around one of our bad guy broken X chromosomes, right? She's not colorblind because she's got a normal one to read, but her father had the disorder, so she's got that broken one there. All right, let's do the Punnett square. We could have this kid who got this X chromosome here and that X chromosome there. We could have this kid and a Y. We could make this kid. Or we could make this kid. Yeah, this one should have 1, right? There we go. Okay. First of all, when these people go to have kids, let's forget about the colorblind gene for a second and just take a look at Punnett squares if we just looked at X and Y chromosomes. What are the odds of getting a girl? Is this one a girl?
Yes.
Yeah, that one a girl? No, this one a girl? Yes, that one a girl? No, so it's 2 out of 4. In other words, the odds of having a girl kid are 2 out of 4, which is the same thing as saying 1/2. Your odds of having a boy or a girl is 50-50 just because of how meiosis works and how Punnett squares work. Like, it's 50-50 odds that you're going to get a boy or a girl. It's hilarious when you, like, hopefully you don't know this yet, but someday when you go to have kids, and, like, your wife is obviously pregnant, people will say all kinds of cutesy things that, like, if you drink a lot of orange juice, it's more likely to come out a boy. Like, people say the stupidest garbage, and and you just have to, like, smile and nod and be like, okay. Or, like, you're carrying out front, it must be a boy. Like, none of this makes any sense. First of all, there's no way to predict gender from the outside. You can't tell. All the old wives' tales about, like, do this, that, the other thing, and you'll have, like, more of this gender or that gender, all BS. None of that's true. There's no way to affect it. It's like 50-50 coin toss whether you get a guy or a girl. Because numbers, that's how math is. Yeah? Anyway, so that's just boys and girls. Now let's look at what happened with this bizarre colorblind trait here. This would be like if I had kids with somebody that was a colorblind carrier. What do I got? Let's start at the bottom because this one's the least interesting. What kind of kid is that?
a normal boy.
That's normal boy, no colorblindness, right? What kind of kid is this?
A girl.
That's a girl. Is she colorblind?
No.
No, because she's got 1 broken X chromosome, but she's got 1 good one. So she'll just read the good copy. How about this kid? That's a boy. Is he colorblind? Yes. And then this one, that's a girl. She's got a broken X chromosome and another broken X chromosome. Does she have an unbroken one to read? No, so this is the only way a girl can get forced into reading a broken chromosome is that she's got 2 of them and they're both busted. She's going to produce abnormal cone structures. She'll be colorblind. So we can get colorblind girls, but you'd have to have a situation where a colorblind guy and somebody who had some colorblind genes who's a girl had kids together. You could make a girl that's broken on both her X chromosomes. So for this couple, the odds are 2 out of 4 chance of colorblind kids.
And that's what the chicks with their boys have been sort.
Okay. So if you want to do what are the chances of the boys? Well, let's see here. The chances that the boys will have the disorder, it's still 50% of their boys. Like 1 out of the 2 boys that they could possibly make. Right? And for the girls, it's still 50% because it's 1 out of the 2 possible girls they could make. So if you wanted to ask, like, what are the chances their boys will have it? Well, it's 1 out of 2. What are the chances the girls would have it? It's 1 out of 2. No matter how you slice it, it's 1/2. Right? Because 2 out of 4 is really, if you reduce the fraction there, 2 out of 4 is the same thing as saying 1 out of 2. Right? And 1 out of 2 is 1/2 or 50%. These people have 50% chance of producing colorblind children. Ooh. I was saying that I already know stuff about like my kids, my daughter, whatever, and her odds of passing on colorblindness. So this is, I'm changing the Punnett square a little bit here. But this is like when my wife and I went to have kids. So here's me, here's my wife. When we went to go make kids, we made that kid. We can make this kid. We can make, this kid. Now, because I'm a biology teacher, we had 1 boy kid, 1 girl kid, just so they'd make a good example for this problem. That was the whole thinking behind why we had 1 boy and 1 girl. I'm kidding, it's just random chance. Anyway, but this is what happened with my kids. Like, I can tell there's no way, it's not possible that my daughter could have colorblindness because she did get 1 X chromosome from me, and she had to get the busted X chromosome from me because I know she's got 2 X chromosomes, 1 for my wife, 1 for me, because she came out a girl. So 1 came from me, so I know she's a carrier. I know my daughter's a carrier. But my wife has absolutely 0 family history of colorblindness, so she must be passing on normal X chromosomes, so that's what's going on there. Can my boy kid, my son, can he possibly be colorblind? Where did his only X chromosome come from? Am I giving him an X chromosome? Nope. I gave him Y chromosomes. That's what made him a boy. My wife gave him his X chromosome, and she doesn't have broken X chromosomes. So my boy can't be colorblind, and my girl can't be colorblind. But 50% of my daughter's boy children are going to be colorblind. In other words, my daughter will have a 1/4 chance of making colorblind grandkids for me. Although she won't, because she already said she's not having kids, and she's just going to live in a house full of pigs. She wants pigs. Just what I always wanted was grand pigs. Awesome. Anyway, hopefully she'll change her mind when she gets older. I don't know. But anyway, that's how sex-linked traits work. You just use the modified Punnett square, and you can easily solve a sex-linked trait problem. It's not really any more complicated than a basic Punnett square problem. You just need to know when to use which one. If it's any other type of genetic practice problem, you're going to use just basic Punnett square without chromosomes on it. If it's a sex-linked trait, then you got to start with your 2 X chromosomes for a girl and your X and Y chromosome for a guy. Because with sex-linked traits, the gender of the kid matters. Good? All right. So, can girls get sex-linked disorders? They definitely can. They just have to pick up 2 broken copies rather than just the 1 broken copy it would take to give a guy a sex-linked disorder. So it's far more rare. Girls, can girls be colorblind then? Colorblind is a sex-linked trait. So can you come across a colorblind girl? Yeah, you can. It would just be really rare. I made sure to mention hemophilia in there because that's probably the most common. Hemophilia is probably the most common sex-linked trait that's actually like deadly. If you're born with hemophilia, your life expectancy is much, much, much shorter. Partially because of the bleeding risk that I already talked about, like people with hemophilia, they don't clot their blood normally. But also that makes it so if they cut their skin, their cuts stay open. And if skin cuts stay open for a long time, then you increase your risk of picking up bacteria and bad guys, like pathogens from the environment. Bacteria and viruses get in easily through open cuts. If you have normal clotting factor, you form a scab right away, and that protects you from bacteria and stuff getting in. But if you're a hemophiliac, you don't make scabs right, and bacteria and viruses get in you. And typically people with hemophilia eventually die of hepatitis because the hepatitis virus it lives on like every surface you ever touch. Like it's all over the place all the time. You get exposed to it all the time. Luckily, you have skin that keeps it out of you, but if you have hemophilia and you get cuts in your skin, it doesn't clot right, you get hepatitis, and hepatitis eventually kills you. So that's sad. I had 2 different friends in college that had hemophilia, both of whom are still alive and well. So, yay. Treatments are way better than it used to be. Used to be that people with hemophilia, they had a lot of internal bleeding issues and they were in and out of the hospital all the time. Now you can live a pretty normal life with treatment. And soon, what they're going to be able to do is go in and fix it. Well, I've said it a million times, we're at the dawn of genetic medicine. They've already identified the genes that give you hemophilia. When we get to the point where we can do gene editing on whole live humans, which is coming here in the next 5-10 years, we'll be able to cure hemophilia. Like right now we just treat it, but in the very near future we'll be able to cure it. So that's neat. Okay, last thing I want to cover today real quick is incomplete dominance. And basically the only reason I want to teach you about incomplete dominance is to drive home the point how incredibly rare incomplete dominance is. Most people on the street, if you just went up to them and asked them, how does genetics work? Most people think genetics works by mom and dad mixing traits together to give like a kid that's somewhere in between. That's not really how genetics works. Most genes come in either dominant or recessive form, and both parents contribute a gene, but the dominant one gets read, the recessive one gets ignored, right? If there is a recessive gene. That's really how genetics works. But occasionally you run into weirdo genes where some of the recessive trait shows through. Those genes that have a recessive trait that don't get completely covered by the dominant, those are extremely rare. So even though most people think that incomplete dominance is the normal way that genetics works, it's not. This is a really, 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 really rare version of inheritance. So Mendel looked at pea plants that had really simple genetics, and some rare genes, a dominant trait won't be completely dominant. It won't completely cover the effect of the recessive. These are extremely uncommon. Every biology textbook I've ever seen, which is a whole bunch of them, they always talk about these flowers. I want to say that they're pansies, I think, is the name of the I don't remember. It doesn't matter. But there's this one rare breed of flower that has either red offspring, or it produces either red flowers or white flowers. And if you breed a red one with a white one, you get pink offspring. Yeah? Carnations do that. There might be several different flowers that do that. I don't know. But that's rare. That's weird. The fact that every biology textbook uses the same species of flower and shows that, like, hey, sometimes this happens, it's because it only happens in that flower with that one particular trait. Or I know that Palomino horses do the same thing. You get a Palomino horse by an incomplete dominant situation with horse coat color. But like those are the only two examples I can think of. All the rest of the genes, when you get like in-betweeny stuff, it's not because of incomplete dominance, it's because of polygenic traits. A lot of your traits are controlled by more than one gene, but each of the genes are Mendelian. Incomplete dominance is extremely rare. Just in flowers and horses, that's about the only time I ever see it happening with anything. It's called incomplete dominance. It's extremely rare. The only reason I want to teach you about it is to make sure you understand this is not the way that 99.9% of genetics works. And most people think this is how all of genetics works. And most people are wrong. Okay. Can I turn page? I want to show you pretty pictures. Oh. Look, those are horses. I know virtually nothing about horses other than I don't like them and they don't like me. Every time I've gone to ride a horse, it's ended badly. All 2 times. Anyway, I know very little about horses besides a little bit of genetics, and I do know that if you want to make a Palomino horse, a horse with this like in-between coat color, what you do is you take a chestnut horse and you take a Carmelo horse and you breed them together. If you look at this chestnut horse, his genotype is abbreviated capital D, and this chestnut, or the Carmelo horse down here is his genotype is abbreviated little d. So when they went to go make babies, this horse would donate capital D. This horse would donate lowercase d. You'd end up with a heterozygous offspring horse. If this was a normal Mendelian gene, what color should this horse be? If it was normal Mendelian gene. He's got capital, lowercase. He should just look like this one, right? The dominant should be the only trait that gets expressed. But because this is one of these weirdo examples of incomplete dominance, some of the recessive shows through in this horse, and you end up with this Palomino horse, which is not usually what would happen. But again, I'm just bringing this up because I want to show you that incomplete dominant traits are extremely rare, and you have to go to some pretty bizarre, like there's not any good examples of like external features on humans that are controlled by an incomplete dominant trait. It's just not a thing that happens in people very often. It's not a thing that happens in horses very often either, except for this one coat color example. Like, that's about it for incomplete dominant traits. So make sure you know incomplete dominance is extremely rare, and there's only a few examples I could point to, and horses is one of them. Yeah. I don't know. I don't understand. What's the difference between a chestnut and a bay horse? Look like the same
Chestnut is technically red and a bay brown.
Okay. You know more about horses than I do, because I would have just been like, that's a brown horse and so's the other one. And then I'm colorblind, so like brown and red, that's the same color. Once in high school when I was filming Spanish skit, a horse walked up to me and bit me. It's awesome footage. I got to find it somewhere in a VHS tape. And it just walks right up to me, just chomp, like no reason whatsoever, totally unprovoked. And then when I used to work at a water ski camp, and we would go steal the horses to go to the other side of the lake. We'd go to the horse camp and steal their horses. It was fun. Because the other side of the lake had better water. Like the wind came over the lake, and like the far side of the lake was the smooth side. So anyway, we liked the far side of the lake, but to get all the crap over there, water skis, a bunch of kids and stuff, we'd steal the horses. But then I was going so we're going to steal the horses, and I put all the horse stuff on the horse, saddle and the crap that goes on the horse. And while I'm putting the stuff on the horse just like did like sideways walk thing and just like crushed me into the side of the horse garage thing. It just like walks into me and then kind of like looks back over its shoulder like, hey, look, I can smash you. Like, neat. I don't like you. It's a problem with horses. I don't like vehicles that have their own brain. They usually go where you point them. Usually. But then sometimes they're like, no.
My horses don't listen to me at all.
Yeah, but they're a little bit like a dog, except they're like 1,000 pounds. They can really damage you. I don't trust them. I like to have brakes. And a motor. What I really want is a motorcycle. It's a much better idea. You don't have to feed motorcycles. That's not true.
Oh, my horse, well, one of them, he eats 17 pounds of grain a day.
Oof.
Grain.
17 pounds?
Yeah.
So like I buy my dog like a 55-pound bag of dog food, and that lasts like a month. You gotta be going through food just like... How much does a horse weigh? I really have honestly no idea. Like, they're a little over 1,000 pounds, yes?
He weighs like 1,600 something.
Oh, there's a lot of animal.
Yeah.
Horses are terrifying. Anyway, I'm glad I don't live in horse and buggy days. Motorcycles were invented for a reason. Anyway, sorry if you take offense. I don't get along with horses, but I've known plenty of people that actually enjoy horses, and like, good for you. They just don't like me. I don't like them. We stay away from each other. Let's not do these yet. This is going to be an example when we get to this part. When we get more than 2 alleles in a population, that can happen. That's going to do some weird things to genetics, but I'm going to leave some stuff for later on this week. We're already over 35 minutes on this thing, and I had to fight with Zoom this morning, so we burned some time. We don't have time to get through this next part. I'm going to call it a day right there. Please, again, I know I've reminded you 8 times, but the end of the marking period is on Thursday. Please make sure that your grades are where you want them to be and get everything taken care of well before Thursday so I've got time to get it in the grade book. Cool? Got it?
Yeah.
Everybody happy with everything?
Yeah. Yes.
Neat. Good. I'm done.